If you like this video, please subscribe. Thank you. Phospholipase C is an important intracellular enzyme that is activated by a G protein coupled receptor and can produce two potent intracellular second messengers. There are 6 important features about phospholipase C and its activation that are worth noting. The first feature has to do with its substrate, phosphatidylinositol 4,5-bisphosphate, abbreviated PIP2. This substrate resides in the inner leaflet of the lipid bilayer membrane of the plasma membrane. Second important feature of phospholipase C is how it acts on its substrates. Phospholipase C, when activated, will hydrolyze its substrate PIP2 in the plasma membrane by breaking the bond between the glycerol diacylglycerol backbone and the phosphoinositide head group, thus releasing 2 new molecules. The 2 new molecules are diacylglycerol and inositol trisphosphate. The third important feature of phospholipase C is that it is activated by the activation of a specific cell surface receptor. Notable among these receptors that will activate phospholipase C is the alpha-1 adrenergic receptor, which is activated by epinephrine or norepinephrine. The fourth important feature of the activation of phospholipase C is that receptor-mediated activation is mediated by the G protein alpha subunit designated GQ. When the alpha-1 adrenergic receptor is activated by epinephrine, it will activate the trimeric G protein GQ. This activation is followed by the dissociation of the alpha subunit of GQ from the beta gamma subunits, and the replacement or the exchange of GDP bound to the alpha subunit by GTP. The result is that G alpha Q bound to GTP is now in the active form and is capable of activating phospholipase C. The fifth important feature of phospholipase C is that when G alpha Q with GTP bound to it associates with phospholipase C in the membrane, it will activate it and cause the hydrolysis of PIP2 in the inner leaflet of the plasma membrane, producing 2 two new molecules: diacylglycerol, which remains in the plasma membrane, whereas the other molecule, inositol trisphosphate or IP3, is released from the PIP2 molecule in the cytoplasm of the cell. Thus, the activation of phospholipase C initially by GQ alpha initially produces two second messengers that were absent prior to the activation of the enzyme. These second messengers are diacylglycerol and inositol trisphosphate. Inositol trisphosphate in the cytoplasm eventually makes its way to the endoplasmic reticulum, where it can bind to specific receptors and release stored calcium from the ER or the endoplasmic reticulum. As such, activation of phospholipase C produces 3 second messengers in total, whose amounts are going to increase as a response to this activation. And the 3 second messengers produced are diacylglycerol, IP3, and calcium. The sixth important feature of phospholipase C activation is the cellular responses it can produce. The first cellular response produced has to do with the rise of intracellular calcium that in of itself can produce a variety of intracellular responses. The second important intracellular response is that both calcium and diacylglycerol that is still in the membrane can synergistically together bind to an important protein kinase designated protein kinase C. Protein kinase C is then activated due to DAG and calcium binding to it, and which in turn increases its activity and allows it to phosphorylate specific intracellular target proteins / enzymes, changing the respective activities of these proteins or enzymes either positively or negatively. If you like this video, please subscribe. Thank you.